Hello and welcome to Lecture 3, Animal Development. There are two distinct parts of this one PowerPoint, so I recommend going through the first half and then taking some time learning that material and then going through the second half of the presentation. So the first half is regarding Chapter 8. So in part 1 of this presentation, we're going to talk about the stages of development. Animal development occurs in various sequential stages, and they are also in a hierarchy system. So as you go through time or as you progress through the stages, the organism becomes more and more complex. And each prior stage facilitates the development of the next stage. So it sets the environment and the tone for the next stage. The main stages of development are fertilization, cleavage, gastrulation, organogenesis, and then growth. So first, fertilization. Fertilization is the union of male and female haploid gametes to form a diploid zygote. And this is referring to sexual, not asexual reproduction. And this is the fusion of sperm and egg nuclei, pronuclei. After the nuclear fission, the egg cell is no longer called an egg, it is called a zygote, and it is now a diploid zygote. Fertilization, in addition to fusing the nuclei to begin the process of making a zygote, it also activates the cytoplasm. So different processes will start to occur within the egg that were dormant before. This includes DNA and protein synthesis, and this is to allow the cell to start dividing and replicating. And then also there's a spatial rearrangement of the components within the cytoplasm so that as the cells are dividing, depending on the type of organism that it'll be, it can start to develop properly. After fertilization, you have cleavage. And cleavage is when you have your large egg cell, which, mind you, is significantly larger than the average somatic cell, will then undergo cellular division to become 2 cells, which each of those cells will then undergo cellular division and become 4 cells. And each of the resulting cells from a division, so both this cell and this cell, are called blastomeres. And during this division process, there is no growth in these cells. So the egg cell is quite large, and it just keeps dividing and dividing and dividing and dividing until each of the blastomeres is approximately the size of a normal somatic cell. This will require potentially hundreds to thousands of divisions until the large egg cell is split into enough small blastomeres to form something that we call the blastula. And the blastula is a fluid-filled ball of cells, and essentially that middle fluid-filled area is called the blastocele, and it's surrounded by an outer layer of single outer layer of cells. And that outer layer of cells will actually go on to become the ectoderm in the resulting animal. All animals have a blastula stage of development. After cleavage, we have the gastrulation stage. And this is when one side of the blastula will begin to invaginate and form this pouch within the blastocele. So in the image below, this area down here of the blastula has started to push its way into the blastula and forms this kind of U-shaped pouch. This is called the archenteron or the gastrostele, and it will go on to form the endoderm. So when you have at the gastrulation stage, we now have 2 germ layers, the ectoderm and the endoderm. All animals undergo a gastrulation stage with the exception of the sponges. So sponges stop at the blastula stage, but everything else keeps going to form a gastrula. One very important part of the gastrula is the blastopore, and the blastopore is the opening of the gastrocele or the archenteron here. And it's important because in some organisms, it will later become the mouth of the organism, and these are classified as classified as protostomes. And then, or it can become the anus of the organism, and these are deuterostomes, which include us. If an organism's developmental stage ends here with this gastrula having just a pocket, then those organisms are considered to have an incomplete gut. Then these include like jellyfish, something you might be familiar with, anemones and things like that have this incomplete gut. However, if the gastrula keeps pushing through, so if this archenteron keeps extending all the way to the other side of the ball of cells and creates this tube, this is now the complete gut. So in the gastrula, if you just have this pouch, this is an incomplete gut. Food goes in, food must come out of this side. However, if you have a tube through, it's a complete gut, which is something like we have where food goes in one side and out the other side, okay? And the way I like to think about this process is those jelly-filled toys that some of you guys might be familiar with as a kid, where there's like a hole, a tube through the middle you put your fingers through. It's kind of like that at this point. There's cells on the outside that are the ectoderm, and there are cells on the inside that are your endoderm, which will become your gut lining. And then there's fluid that's hanging out in the middle, which is the blastocele, right? Some animals in their development kind of at two germ layers, so they'll just have the ectoderm and the endoderm. However, the vast majority of animals go on to create a third germ layer, which is called the mesoderm. The mesoderm is a tissue layer that's derived from endoderm cells, and the animals that possess both the endoderm, ectoderm, and mesoderm are termed triploblastic. There are three types of triploblastic body forms. The first are acoelomates. And so these are organisms whose mesoderm completely fills up their blastocele cavity. So there's no space, no air. And then you have coelomates. And coelomates have a fluid-filled cavity that's surrounded by mesoderm within their body. And this coelom, or this fluid-filled cavity, helps to cushion the gut and protect it from harm, physical harm, pressure. If you get punched, it doesn't just rupture, right? So within coelom, the animals that have coeloms, there are two types. There are those that are pseudocoelomates, and so they essentially have a coelom that is kind of one-sided. So it's the coelom lining is touching or is surrounded by ectoderm, but there is no mesoderm lining that is on the endoderm side. And then you have those that are true coelomates, which we just call them coelomates. And these are organisms whose coelom is surrounded completely by mesoderm. And so the different germ layers will later become different things, right? So the ectoderm will become your skin and your nervous system, your neural connections. The endoderm will become your gut, and then the gut lining, and then your mesoderm then becomes pretty much everything else. It becomes your circulatory system, your skeletal tissues, your muscle tissues. All of that is derived from mesoderm. Within the coelomates, there are two ways that a coelom can form. There's the schizocilous and enterocoelous methods. So in schizocilous organisms, the mesoderm, as we mentioned before, is derived from endoderm, and it is in both cases, but here we're going to just talk about this first. It's derived from the endoderm, so you'll see mesoderm here starting to develop, and it will start to fill up the blastocele. And then at a certain point, the cells that are that make up the mesoderm within the middle of it will split. And then due to programmed cell death or apoptosis, they'll develop this kind of space, this gap here, okay? and then that gap will then later become filled with fluid, and that's how they derive their coelom. This is more characteristic. This is characteristic actually of protostomes. This is not characteristic of deuterostomes like humans. What's characteristic of deuterostomes is the enterocoelous method. So this is when, as your archenteron is invaginating within the blastocele, right, which is yellow here, as this is occurring, and the archenteron or the endoderm cells here will start to actually extend laterally, and you see these little pockets here. As they're extending laterally, that is what becomes the mesoderm, okay? And it'll continue to pull out, pull out, pull out, pull out until they pinch off. And you can imagine if they're coming out like this and they pinch off and there's fluid in the center, now we've got our coeloms on the side. So that's how we develop coeloms in this case. Welcome to part 2. So in this part, we're going to mostly talk about cellular differentiation. Cell differentiation is especially important because, as we all know, every cell in your body has the same DNA. But if every cell in your body has the same DNA, how do we get so many different cell types? That's through the process of differentiation, clearly. So differentiation can be stimulated by a couple of different interactions. The first that we're going to talk about is cytoplasmic partitioning, and this is based on where components are located, or where different macromolecules and proteins and things like that are located within the egg cell. Also inductive interactions, so the interactions between a undifferentiated cell and its neighboring cells will stimulate a certain type of behaviors in the undifferentiated cell. And then also gene expression. Gene expression plays a major role in what a cell does and how it will develop. One very important term that we'll use throughout this talk are morphogens. And so these are molecules that diffuse within an environment that initiate some sort of cellular response. So these can be factors that are within the cell, which we talked about in cytoplasmic partitioning. It could be compounds that are within the cytoplasm of the egg cell, or they can be external factors. So they can be chemical signals that are released from nearby cells that induce a reaction in the target cell. The morphogens help to drive early stages of differentiation. So they play a major role in whether a cell even begins to start to morph into the type of cell that it should be. Morphogens tend to have a concentration gradient. So you can imagine if I'm a cell and I'm putting out all types of chemical signals, and there's a cell that's 2 feet from me, a cell, human, who's 2 feet from me, they're going to experience a higher concentration of those chemical signals I'm sending out than someone who's 15 feet from me. Okay, so there's a concentration gradient of morphogens from their release point to everywhere else in the environment. Okay, and yeah, okay. Once a cell is differentiated, there is no turning back. Okay, so undifferentiated cells can become differentiated. So a stem cell can then become a epithelial cell, a nerve cell, a bone cell, etc. But a differentiated cell will not then become an undifferentiated cell. So muscle cells will not go back to become stem cells. Once you're differentiated, that's your fate. So first we're going to talk about cytoplasmic specification. And this is where the cytoplasmic contents of the cell plays a role in the fate of the resulting cell. Okay, so the egg cell has a variety of transcription factors, proteins, mRNA, nutrients within the cytoplasm, but this is not spread homogeneously. So in this image here, that's what they're trying to show you, that in this egg cell, there is a section of the cell that has one type of content, another section has different types of contents, and a third and a fourth section. Right? They all those four sections have different contents within them. This is very, very important when it comes to cleavage, right? So as the cell begins to divide, the resulting blastomeres will have different cytoplasmic contents, and those cytoplasmic contents will then specify what that cell can later be. This is called mosaic development, and it's very common in protostomes, where each portion of this blastomere will then start to develop independently. So they can't develop an organism in isolation. So you can't take this blastomere and separate it and it start to develop an organism on its own, because it has very specific instructions to become a very specific type of cell. This is very different from what we'll talk about in a minute, where in deuterostomes you can do that. But in protostomes that undergo this, that have this type of element of differentiation, each of these cells will have a different cytoplasmic content, and that will tell that cell exactly what kind of cell it will be later. So this cell will be a muscle cell, this cell will be a nerve cell, this cell will be, I don't know, an eye cell, right? And they can't change that. So in addition to cytoplasmic specifications, we have conditional specifications, and this includes inductive interactions. And that is when the fate of an undifferentiated cell is determined by factors or interactions with neighboring cells. And this can be facilitated by the presence of inducers or morphogens, or chemical signals, or through cell-to-cell contact, right? So in the image I drew here, cell 1 is releasing inducers that will then go bind to proteins present on the target cell. Also, cell 2 is releasing chemical compounds that will then bind to proteins on cell 1. Okay. You also have direct contact between cells, where proteins on the cell membrane of cells 1 and cell 2 will then interact with one another, and that can also induce a response in both of those cells to behave differently. So in the case of this example, let's say cell 2 is an undifferentiated cell and cell 1 is a muscle cell in your leg, and cell 1 is putting out all these chemical signals saying, hey, I'm a muscle cell in the leg. That will then induce or cause cell 2 to start to differentiate into a muscle cell within the leg. This provides positional information as well for the undifferentiated cell, as the differentiated cell knows what kind of cell it is. It knows what kind of cell and where it is. So this information will be passed on to the undifferentiated cell. This is very similar to sequential specification, which is essentially the same thing, but instead of morphogens fusing between two separate cells, it's the diffusion of morphogens within a multinucleated cell cytoplasm. This process of conditional specification via inducers and cell-to-cell contact is very common in deuterostomes. Stages. As we mentioned a couple of slides before, there's pattern formation, a determination of position in the body, and induction of limbs and organs. So first, talking about pattern formation, the first stage is identifying where in the body you're located, right? So the body has several axes. You have your anterior, posterior, your proximal distal, and your ventral and dorsal axes. So it's very important in the early stages of development to know where in the body as a cell you are located. And the pattern formation is heavily determined by morphogen concentration. So as I mentioned before, morphogens are molecules that diffuse within an environment, and they have an effect downstream on what a cell will do in transcription of different genes. And so there are many genes involved in pattern formation. We're going to only talk about a few in the next couple slides. So just as a review, in the beginning we have our zygote, which occurs after the fusion of the nuclei from the sperm and the egg. And after that fusion, we get our diploid nucleus. And then that nucleus will begin to replicate over and over and over again to give us a single large cell with multiple nuclei in it. And this is termed the syncytial blastoderm. And as you might remember, a couple slides ago, we talked about syncytial differentiation, and this is relevant here. So in syncytial differentiation, there was a single cell, multinucleated cell that has a differential concentration of morphogens in that cytoplasm. So this is kind of what we're going to talk a little bit more about right now. So within this syncytial blastoderm, in addition to multiple nuclei, we have a lot of maternal-derived morphogens in the form of transcription factors. Two that we're going to talk about in a little bit more detail is in the case of Drosophila, the fly, is bicoid and nanos transcription factors. And bicoid transcription factor, it will indicate the anterior region of the animal, and nanos indicates the posterior region of the animal. And they occur at different concentration gradients throughout the zygote. So if we're looking at this image here, now we have all of our nuclei that were within our syncytial blastoderm have started to polarize themselves and locate themselves on the periphery of the zygote, and they're going to start undergoing actual cell division and differentiating themselves into individual cells. But during this process, you have a various gradient of those morphogens within this region as well. So I'm going to try to draw them in two different colors. We'll say we're going to do red for bicoid. So within this the cytoplasm here, we have high concentration of bicoid at one end, which is represented by those red dots. And as you kind of go through, you get a lower concentration as you travel to the opposite pole of the zygote. Okay, and then the same thing for bicoid. Bicoid will draw in blue. So the opposite end of the zygote cytoplasm will have a high concentration of nanos, and as you travel to the other pole, the concentration of nanos will decrease. So at one end, the concentration of bicoid is high, and as you travel to the opposite pole, it becomes lower. And on the opposite pole, the concentration of nanos is high, and as you travel to the opposite pole, the concentration decreases, right? So this will play a role in what genes are expressed in the resulting cells on both poles of that embryo, of that zygote. So these genes, if we had our cell, this gradient will give you something that looks like this. We have a lot of red, and then it kind of tapers off, right? And then we have a lot of, which is our bicoid. We have a lot of nanos, which is our blue, and it tapers off. And in the middle, we've got kind of mixed concentrations of both. So if you see from that image that I kind of drew here, it now have three segments, and these three segments will eventually become our head, our thorax, and our abdomen. Okay, so and those three regions develop because the bicoid and nanos will start enhancing the expression of genes for their respective regions, and decreasing the expression of genes for the opposite region. So what I mean by that is the bicoid transcription factor will upregulate or allow for the transcription of genes that are involved in head formation or being the anterior end of the organism, and it will downregulate any genes that code for the posterior end, which would be normally regulated by nanos. On the opposite pole of the organism, where nanos concentrations are high, any genes that need to be expressed at the posterior end of the organism will be upregulated, while genes that will normally be expressed at the anterior end of the organism will be downregulated. And then the middle area is where the concentrations are usually lower, but you have a little bit of concentrations of both. So the genes that are impacted by bicoid and nanos are called gap genes. And that's how those gap genes are how we get these three distinct regions. Gap genes are responsible for the initial kind of segmentation into the head, the thorax, and the abdomen. Then after gap genes are activated, which are also transcription factors, those transcription factors will regulate and activate pair rule genes. So pair rule genes will take each of these segments, the head, the thorax, and the abdomen, and further segment them. And then pair rule genes are also transcription factors, and they will regulate segment polarity genes. And those will then take even further segment the organism and identify each of the anterior, posterior regions within the segments that are within the segments. So that can be a bit confusing. When I was an undergrad, this concept was a lot for me. So I've included several videos at the end and also some helpful tips and some resources from the book that might help to explain this and then the other concepts we're going to talk about in a minute. So the next stage when you're looking at gene expression's effect on differentiation is looking at where in the position in the body you are. And so the genes that play a major role in this are termed homeotic genes. And as we mentioned, they regulate the expression of other genes. So they are transcription factors. This whole process, you probably noticed, we've talked a lot about transcription factors. Transcription factors play a major role in the development of an organism and kind of laying out the body plan. Homeotic genes make sure that only certain genes are expressed at certain segments of the body. So after you've designated the body plan, you want to make sure that only certain genes are expressed in certain regions and certain genes are turned off in certain regions. All homeotic genes have this 180 base pair conserved sequence that is somewhere within their region. And this 180 base pair sequence codes for the DNA binding domain of those transcription factors. So this is what allows them to anchor to their promoter region and gets you the expression of your target gene. The homeo box is not exclusive to homeotic genes. However, it is very, very prevalent in them. All the homeotic genes have a homeo box. So we're going to spend a little time talking about HOM genes, or you might have heard of them termed as Hox genes. In Drosophila, they're referred to as Hox genes, but in general, HOM genes, or homeotic genes, are located on various chromosomes within the organism. So they can be located on one chromosome or on multiple chromosomes, depending on the organism. And they help to differentiate the anterior-posterior axis during later stages of development. So like I mentioned, they occur in clusters on the chromosome. And the interesting thing about these Hox genes is the order that they occur on the chromosome corresponds to their area of expression within the organism. So, for example, here we have lab. Right? So all of these genes are present in every cell of the body, right? But we don't want them to be all expressed in every cell. So if I'm a cell that makes up this little cleft area, I want to express lab. Okay. But I don't want to express DFB or ANTP or ADBB because those are for different segments of the body. So lab is expressed hit the head region to help make this hook. However, if we looked at Ant P is expressed in the abdomen region of the embryo, and it's expressed here, but it's not expressed in the head, and it's not expressed in the thorax. And same thing with ADBB or ABDB. It's expressed in the thorax cells, but it's not expressed in the head cells or the abdomen cells. And this helps to orient the cells and tissues to where they are along the anterior posterior axis. If I'm a cell and I am experiencing high concentrations of lab as a morphogen out there and high expression, I'm getting signals that tell me to express lab, right? And I have that in my environment. I think, okay, I'm a head cell, I'm supposed to make this hook. If on the opposite end, their expression of ABDB is being upregulated, then I'm likely in the thorax. So Hox genes play a major role in kind of locating where exactly along this anterior-posterior axis are you located, what kind of cell should you be. The final stage is making sure that limbs and organs are formed in their appropriate position. So if you're an organism, you want to make sure that you're properly developed and everything is where it should be. If you're a human, you don't necessarily want, you definitely don't want an arm where your leg should be, or you don't want an eye in an unnecessary space, or your organs to be on the opposite side of your body, right? You want to make sure every limb and organ is in the proper orientation. And to ensure this, Hox genes, morphogens, and the body plan layout all play a major role in making sure that limbs are produced where they're supposed to. So we're going to take a look at this image. It's like a chicken embryo, and this is the apical bud to make a wing. So the body plan, as you can see in this kind of cross directional image, the purple region is anterior, right? Because these axes have already been determined. The anterior, posterior, proximal, distal, and then the dorsal ventral axes. And so these cells here know to become a wing and not a leg because of the Hox genes that are being expressed in that area and the morphogens that are being produced in that area. And those are indicators that say, hey, you are in the anterior region of the organism, and you need to be some sort of limb. And so when you put that information together, all the information together, it will form a wing rather than a leg. And this is very important because morphogens can be the same in various parts of the body, but what Hox genes are present, which will tell you what kind of transcription factors are being expressed in that region, will dictate what kind of limb that those cells will become.
Animals share an evolutionary history, and this can be seen no better than in developmental biology. So most post-gastrula vertebrates, so animals that undergo a gastrulation stage and they have a neural nerve tube, a spinal cord, show similar developmental characteristics, which ultimately suggests that there's a shared evolutionary history. So when you look at the image here, it's pretty apparent, right? In the beginning, these embryos aren't very different in appearance, right? It isn't until they start to continue to grow and develop that we start to see that these organisms are not actually the same animal, that they represent a fish, a salamander, a tortoise, a chick, and a human, right? And they look similar because they share some key characteristics, and this is among chordates, right? So they have a spinal cord. All these embryos from the fish, salamander, tortoise, chick, and human all have a dorsal neural tube in their embryos, a notochord. They have pharyngeal gills, pouches at some point, so you can see that here as these kind of bumpy, bubbly things are their pharyngeal gills. They have a ventral heart and a post-anal tail. So, including us,
We had a tail. You can still kind of see some of the remnants of the tail, right? So with that said, we have reached the end of our learning for this chapter. This completes chapter 8. I recommend going and reading chapter 8, sections 1 through 7, and completing the Smart Book assessment as well. This is a very complicated topic for many students, so really take your time kind of going through it and learning this material and seeking out resources that might help to explain this material. Have a good day.